Hi everyone, this is our lecture dealing with a couple of topics from Chapter 5 in the textbook, and again this is for Section 1, which we'll be having our exam on Thursday. I wanted to go over a couple of these topics that we didn't really have time to fit into a POGL assignment. This is dealing with 5.1, 5.3, and 5.4. The part from 5.1 you will be expected to know how to calculate. It's a pretty simple formula. Whereas the parts from 5.3 and 5.4, at this point in the class, you're not really going to be expected to use those formulas to calculate anything, but I want you to know what they are, what they're for, how people use them, etc. So with that, I'll start the video. So in Chapter 5, a lot of what we're talking about is the matrix. As I'm kind of surprised we haven't spoken about the matrix much at this point, but we've really been sort of talking about hypothetical ideal solutions. So what we're talking about in terms of chemistry, when we're talking about the matrix, what we're talking about is the matrix is the chemical environment that your sample resides in. Right. So this is, if you think about it, if your samples, we're typically dealing with aqueous solutions. So what ends up happening is that if you make a nice neat lab or nice neat sample in the lab, your matrix may be really simple, and that matrix may not influence your measurement at all. But if you're using a really complicated sample, as we're going to see later throughout the semester, like for example in the semester we're going to be doing some experiments where we extract a couple of compounds from chocolate, and chocolate has a lot of other gunk in there, a lot of other chemicals in there, and that can actually affect your measurements. So I want to show you this example right here. This example is something that I pulled out of literature, and this is for this drug called olanzapine. And so somebody did an analysis where they showed, so this is sort of a neat spectra of taken from olanzapine, and then olanzapine when the same amount of olanzapine if they put it inside the matrix, which is sort of a biological matrix. In this case, this came from cell lysates, so they just had it in some cells. They broke up some cells, and the contents of those cells actually influenced the detector response. And if you look at this, the maximum response here is around 120 intensity. This is sort of an arbitrary unit. Whereas if you look at the sort of neat effects, or the neat signal in a really uncomplicated matrix, see this is around 64,000. So it's a pretty huge effect can happen, and this can actually really influence your data. And so there's a couple of ways we deal with this. One way is using this percent recovery. So this is a way of really diagnosing your matrix effects. But this really goes beyond matrix effects and can be used for any sort of sample process, trying to figure out how much of your sample you're losing along the way. So for example, if you do like a 12-step process where you're extracting and you're filtering and all that sort of stuff, you could end up losing some of your sample along the way, and you could also use the same equation for that. So this is just called percent recovery. All right, so what you have is, the way you do this is that you actually take your sample, you take two measurements of your sample, you sort of split it into two, and one of your samples you add a spike, which is a known amount of compounds, so that's this concentration added, okay? And then you measure both the spiked sample and the unspiked sample. And you sort of figure out the difference between those two and divide it by the amount you added, and you multiply it by 100, this will tell you the percent recovery. What this is telling you is what percent of that stuff that you added is measured in your process. Okay, so this can tell you a bit about matrix effects, because if you know how much you added and your measurement is less or more than that, that can tell you sort of how that matrix is influencing your measurement. So we're just going to go through a quick example here. So let's say, imagine you do a calibration curve, you're running for unknown samples. This is sort of arbitrary here. Your unknown sample contains 10 micrograms per milliliter, or at least this is what you determine it to be. Alright, and then you spike in 5 micrograms per milliliter, so this is your spike, and determine that the concentration of the spike sample is 15.3 micrograms per milliliter. So what we have here is this is our C unspiked. Down here we have our C spiked, and then our C added. So you see, it's actually a pretty simple equation once you figure out what each part is. And so if we set this up, we get our, remember, our percent recovery equals our C of our spiked minus C of our unspiked divided by the amount that we added here. So it's our C added. And this will, and you multiply it by 100. So in this case, from our previous slide, we found that our C spiked was 15.3. So as long as the units are all in the same dimensions, they're sort of arbitrary. And so I'm not going to include them here. 15.3 minus 10.0, this is our C unspiked, if you remember, C unspiked, C spiked, and then the amount that we added was 5.0. You multiply that by 100, you get 106. Okay, that's 6. All right, so you can see here that our percent recovery is 106%. So I just want to show you that matrix effects. So our matrix effects can be either positive or negative. Okay, so in many cases you'll find that your matrix is giving you a lower measurement. Oh, that's not the color I want. Pretty here. All right. Matrix effects are either positive or negative. All right. So, all right. So there's so they can give you either sort of in this case we have an inflated large inflated apparent concentration of our compound. And so if you want to figure out your actual one, you could use this percent recovery to sort of get a good estimate by essentially dividing the amount that you measured by what this percent recovery is. Right? And that would sort of be a way to compensate for it. But our ways to deal with it, you know, there's 3 different ways, 3 different main ways. One way is to right, get your sample out of the matrix. Okay. So in that case, if we get it out of this matrix, right, it's always going to be in some sort of matrix. But the question is, if you can get it out of the matrix, and this is our representation of this, right? This is your sample. It's the matrix, all right? So if you can pull your sample out of the matrix and put it in something, a better environment, maybe something where it won't influence your measurements, then that's a win, and that's the best thing to do. But the problem is you oftentimes can't do it because your matrix may be difficult to remove from your sample or vice versa, and you just may not be able to do that, and so you have to come up with some other method. So the second method that we'll do is called standard addition. And this is one of those processes that is used pretty widely in the lab, and you use this equation here essentially to determine what it is. And so how this actually works is you start with your sample. All right, so this is a picture of, say, so you start with a volumetric flask here, and you add your sample, an aliquot of your sample to each one of these. Okay. And remember, I was sort of saying that there are some examples where you really want to give a precise volume. So what you might want to do is take a volumetric pipette in the same volume of your sample into each one of these. Okay. Then after that, you add a standard, right? And you add them in increasing volumes. And so for the first one, you don't add any. And you, in this case, this example here, you add 5 milliliters to the second one, 10 milliliters to the third, 15 to the fourth, 20 to the second. And then you dilute it to the mark. All right. And so now what you've had is that you've added a standard, and the standard is the same. The standard is the same compound, same as your analyte. Alright, and so what you'd imagine if you measured, if you're doing, in this case, this sort of shows, this is chromatography, which we're going to talk about later in the semester. What you should see is an increasing signal, right? And each one of these signals increases by, should be roughly equivalent to that volume. And you can use this equation here to figure out what your volume should be. So, sorry, these pictures did not turn out looking real well, but these come from your book. This is 5.8. All right. And basically you fit the line, you plot in this sort of funny fashion here, and you fit the line, you find the intercept here can tell you your concentration of your unknown. So this is the example of something that I'm not going to expect you to know how to do for the exam, but just to know what it's for, okay? Later in class, when we're doing an experiment with fluoride with the ion selective electrode, you will be doing this, and so you will have to calculate it at that point. But for this exam, you don't need to know. You just need to know that this standard addition method is used to eliminate the matrix effects, or to compensate for it. But you can imagine there's probably a downside to this, right? And the downside is you have to have a lot of your standard. You have to do a lot of measurements, okay? And so that's may be an issue depending on or a lot of your unknown, a lot of your analyte. And so that may be an issue depending on what your analyte is. You may just not be able to do it, or maybe your analysis is so expensive that you can't just do it this many times. And so another alternative way is this sort of third method. And this is called using an internal standard. An internal standard uses, so this, by the way, this standard addition methods here, this is from Harris 5.3. Okay, and this internal standard method is from Harris 5.4. Okay, and what you do is essentially you use a standard of a compound, which is something, and your internal standard is not your compound. It's not your analyte, but is very similar to it. So the idea is that anything that affects your analyte should hopefully be affecting your internal standard as well. Alright, so this is sort of an example here. You have an unknown x, and you have an internal standard. The idea is you've put in a known amount of this, and so as long as whatever is affecting your analyte, your unknown, is affecting it in an equal way that it's affecting your internal standard, then you can use this equation. All right, and basically you have the signal from your analyte divided by the concentration, right? So this is something that you get from this, right? And then you also can grab the information from your standard, right? And you plug that in here, right? So these are both measured, right? And this standard concentration, you've added this yourself, so this is known, right? And so you just do two measurements here. This one is also measured. Okay. And so basically you plug this in and you solve for x. Okay. And so this number here is just going to be some fraction, some sort of number that you just determine empirically from your experiment. So you can see this is a lot simpler. You don't need to know it. You don't even need to have, because in some cases you can't even get standards of your analyte. You can use something else in its place, and this is a way to do that, and you only have to do one measurement, unlike the standard additions. But the downside is you're making an assumption here, right? You're assuming that whatever's happening to your analyte is also happening to your standard, and these may not always be 100% accurate. One of the best ways to do this is to use an isotope labeled method, and that's where you use just standard isotope label of your actual analyte. They're two chemically different things that can be measured by mass if you're using a mass spec, which we'll do later in the class. That's the way that I do that actually in my lab a lot. But this is another method. Again, I'm not going to expect you to go through and calculate this at this point in the class. Later in the class, towards the end of the semester, you actually will use this formula. So, but I need you to sort of know and understand how these processes work and how you, and under what situations you would use them. So again, this one's from chapter 5.4. The other one here is from 5.3. And this calculation we did right here is from 5.1. Okay. All right. So that's the other stuff that you're going to need to know from Chapter 5. The rest of it we're going to be getting from the POGIL assignment. You guys already started that on Monday, and we'll finish it up on Tuesday. So see you in class soon.